be our last look into the nervous system. For what we just got done talking about in our previous lecture, we're mainly focusing on output, which we describe output going away from the spinal cord or brain as motor or efferent. But in this lecture, we're talking input. So we're talking about signals coming in by way of sensory neurons, providing us with sensations, which involves the afferent nervous system. So let's go ahead and just define sensation. It's actually something that we've defined before. Perception is something that we've also defined before. Defining sensation. This is the conscious or subconscious awareness of changes in the external or internal environment. It's something as simple as being aware that it's cold outside when you walk outside today on this very cold day. We're completely aware of that. We have perception over something like that, which is conscious awareness and interpretation of the sensations. And as we learned in our lecture the other day, with those various functional areas of our cortex, like the primary visual area or the primary somatostatic sensory area, the cortex is involved with perception. Do we have perception over all sensations? We just talked about the fact that for a lot of our autonomic reflexes, things like blood pressure, urination, defecation, which don't really fall in because everyone knows are occurring. But changes in blood pressure are subconscious, and we're not necessarily aware of them. The nature of the sensation and the type of reaction generated will vary according to the ultimate destination nerve impulses, conveying those sensory information, or that sensory information, to the central nervous system. Each unique type of sensation, like touch, pressure, or touch, pain, vision, or hearing, is called a sensory modality, and we classify those modalities into two major groups: the general senses and the special senses. We'll first talk about general senses, and there are two types of generals: somatic and visceral. Somatic come from either the skin, your muscles, your tendons, or your joints. Okay, so within our skin, for example, we have tactile sensations of touch, pressure, vibration, itch, and tickle, as well as warm and cold, which are the thermals. There's pain within our skin, but pain is a sensation that we feel in a lot of places, not just the skin. Proprioceptive sensations are the ones that come from our muscles, tendons, and joints. Things like muscle stretch and muscle or tendon tension, which we've talked about, muscle spindles and Golgi tendon organs just recently. We talked about those on Wednesday. Visceral senses provide us information about conditions within our internal organs, like the stretch of the stomach, the pH of our blood, the O2 levels of our blood, CO2 levels of our blood, many, 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 many things that we'll talk about in this lecture and going forward later in the semester. The last half or so of this lecture goes through the various special senses that we have. There are 5 of them: smell, taste, vision, hearing, and equilibrium, which I guess, I don't know why I put the only one there, equilibrium, with its other name, and which is balance. Where smell is known as olfaction, taste is gustation, vision is just vision, hearing is also known as audition. So there are 5 special senses, and we'll look at those in greater detail. We probably won't get to that today since we already are short on time. Regardless of what the modality is, whether it's pain or pressure, itch or sight, the process of sensation begins in a sensory receptor. Sensory receptors we've talked about since the beginning of the semester. When we talked about feedback loops, remember the first component of a feedback loop was a sensor. We also know them as receptors. Okay, well, you can combine those words and call it a sensor receptor, which are going to be generally one of two things. Either a special cell, which are associated with some of our special senses, or just the dendrites of a sensory neuron. The overall function of a sensory receptor is to convert the energy of a stimulus into the energy of a nerve signal, which then is going to propagate into the central nervous system so that the nervous system, or the central nervous system, can detect and interpret that stimulus acting on or within the body. So it's just to provide input, as we learned earlier in the semester. The process of sensation involves 4 steps. And this is the same 4 steps for something as simple as putting pressure in on a fingertip with a pin or a probe, or something as complex as what we see, or hear, or taste. First of all, there has to be some stimulation. Once again, remember, neurons aren't firing for no reason. They have to be stimulated. So to stimulate a sensory receptor, we have to have an appropriate stimulus occurring within the sensory receptor's receptive field, which is a body region where stimulation activates the receptor and produces a response. I like to think of receptive field as like the amount of area you can cover with your arms spread out wide. Okay. Here we can show what receptive fields are like. Okay. Here on the next page we're showing sensory neurons that have separate receptive fields, versus over here on the right we're showing where 2 neurons have overlapping receptive fields. Some places in our body we have a whole bunch of sensory receptors, and therefore we can describe them as being really sensitive. What places in our body would you think are really sensitive?
Tips of our fingers.
Tips of our fingers.
Our tongue. Or mouth.
Our tongue, our mouth, our lips, sure. Why do you say those places?
Because they're going to be the things that hot, liquid, or sharp objects would touch first.
Touch. That's what we use to touch, right? Okay. Obviously, our special organs, like our eyes and our ears, are going to be loaded up. But when you just talk about like your lips or your hands, fingertips especially, you're putting stuff into contact with those all the time. When you pick something up, you need to be able to process and feel it, right? Okay, well, that means you have to have a lot of receptors to send signals into the brain, okay, or the spinal cord. In other places like the back of our body, you're not picking stuff up or touching stuff with your butt or your back, so you don't need as many sensory receptors. So we don't describe those places as having as much, sensation. So when I say receptive field, I mean basically to stimulate, for example, this singular neuron, we have to stimulate it within its area. Okay? If I were to touch over here where there is not that sensory receptor, am I going to stimulate that receptor? No. So if you think of it like stretching your arms out, and I throw you a ball within the amount of distance you can cover from one arm to the other, one hand to the other, you have the capacity to catch it. But if I throw it all the way over there, and I can't move to go over and catch it, am I going to have the ability to catch it? No. So if the ball in this case is thrown at the width of those dendrites, it has the capacity to stimulate it, but if it's outside of it, it won't. And fortunately for us, in places where we do a lot of contact with stuff like our fingertips and our lips, there are so many sensory receptors that they overlap one another, so the ball's never going to get dropped, if you want to think of it that way. After we stimulate the receptor, then we have to convert that stimulus energy into an action potential. Or for that, really even just a graded potential, a slight depolarization. Before then, we can reach threshold and propagate it inward. That process where we convert energy in a stimulus to a graded potential is called transduction, or transducing, converting the energy. So in this case, our illustration at the bottom is showing the stimulus being a probe pushed in on the fingertip. Inside of our fingertip we have sensory receptors that are stimulated by that probe pushing inward. We're causing depolarization of ion channels. Ions flow in. We create a graded potential. If enough ions flow in, we hit threshold. Guess what happens when we hit threshold? Action potential, right? Okay. Well, since we're going to continuously push that probe in, we're not probably going to just be sending in one action potential. We're going to send in multiple action potentials, which are then going to propagate. Okay, so step 3 there, when the graded potentials in the sensory neuron reach threshold, triggers one or more nerve impulses, which are then propagated to the central nervous system. These neurons then propagate the impulse, or the neurons that propagate this impulse are called first-order neurons. We call them first-order neurons because guess what? They're the first ones in line or first in order. Okay. Remember the nickname for the hypothalamus, or the thalamus, excuse me, was our gateway to the cerebral cortex, right? Look where our sensory signal is eventually going. Through the thalamus, and then from the thalamus to our cortex, which is where we integrate. Particular region of the central nervous system, integrating sensory nerve impulses. Conscious sensations or perceptions are integrated in the cerebral cortex. So things that's like pushing it on your finger with a probe. You're going to be able to feel that and consciously be aware that's happening. Are we consciously aware of every sensation? No. Okay, like when your pH in your blood drops from 7.4 to 7.35. We're not consciously aware. So we don't have conscious awareness of everything. So not everything ends up right out there in that cerebral cortex for perception to occur. I just said converting energy of a stimulus to energy of a graded potential is called transduction. So in our nervous system, converting the stimulus energy to a nerve energy is called sensory transduction. And it is possible as a result of what we call a receptor potential, which is an electrical change in the receptor caused by the initial effect of the stimulus. Okay. In this case, our receptors are the dendrites of this unipolar neuron. Remember, unipolar. So by off to the side, most of our sensory receptors are unipolar. Sensory neurons are unipolar. The dendrites, in this case, are stimulated by a cold stimulus. Maybe we jumped in a cold pool. Maybe we stepped outside on a day like today. These dendrites are activated by the cold stimulus, and they convert that stimulus energy into a depolarization. And if it depolarizes enough, it's going to trigger nerve impulses that are then going to propagate into the central nervous system. Okay. So just basically stuff we've already talked about, which is nice because this is kind of helping you review, hopefully, for Monday. What I'm going to do now for the next couple slides is talk about the different types of receptors we have in our body and the different ways that we classify those receptors. Did you guys have it right? Okay, so first way that we classify sensory receptors is based on what they look like under a microscope. So it's very simply just their anatomical structure. And We can see those three here on the next couple of slides. The first type are called free nerve endings because they are just the dendrites that are uncovered. So free nerve endings are just bare dendrites having no special association with the accessory cells or tissues. Very abundant in our epithelial and connective tissues, which happen to be in our skin, but in other places as well. Examples of free nerve endings include the warm receptors that respond to rising temperatures, cold receptors like what we just saw here on the previous page. This would be a cold receptor responding to falling temperatures. Nociceptors, which are pain-sensitive receptors. I always remember no pain, no gain, or no, I don't want to stimulate those because pain is not fun. And then hair root plexuses, which you take anatomy, you'll learn those are going to wrap around hair follicles, which allows your hair to be an extension and process and stimulate those receptors when you touch your hair or tickle it or pull on it. Encapsulated nerve endings are similar to those bare dendrites, except there's a capsule surrounding it. The dendrites are located inside of the capsule, and the capsule is made up of either glial cells, which remember are the supporting cells of the central nervous or of the nervous system, or it's just connective tissue, functioning to increase sensitivity and specificity of the receptor. Different types of capsules enhance sensitivity or specificity of the receptor. We'll talk about some of those here in a little bit. And then we have what are called special cells or separate cells, which are not neurons, but synapses with them. And these are only found with some of our special senses. Examples include the hair cells that we find in our ear for hearing and equilibrium. We have taste receptor or gustatory receptor cells for taste. And then in our eyes, we have photoreceptors. Anybody ever heard of rods and cones? Rods and cones in your eye are the photoreceptors that provide us with vision. Okay, on this figure here, we're showing a taste receptor. It's stimulated by a sugar molecule binding to it that then synapses with the dendrites of our first order neuron. So this one takes just a couple extra steps. The ultimate goal is still the same, action potential propagated in the central nervous system, but here we have to stimulate this cell, which then stimulates the neuron. Versus here we're just directly stimulating the neuron. With these two, we create what we call a generator potential. With this, we create what's called a receptor potential. These are just two different types of graded or local potentials. Happening in those free nerve endings and encapsulated nerve endings, as well as the receptive part of olfactory receptors, is a generator potential. Okay. Think of these as they actually have the capacity to generate an action potential. So I would think generator can generate. There's not an extra step involved with generator potentials. Once they depolarize enough, they hit threshold, the action potential is carried or propagated. With these special or separate cells, they create a receptor potential, which is what they are, they're receptors, which then, when they are depolarized, will release neurotransmitters just like we saw earlier in last unit. Action potential hits the knob, neurotransmitters are released from the synaptic vesicles. Well, here's essentially the same thing. There are synaptic vesicles located on the side of the cell facing the dendrites, So this receptor potential triggers the release of those neurotransmitters that then activate the dendrites to depolarize, and then an action potential will get propagated. So in this case, we create a postsynaptic potential in the first order neuron, which then can trigger one or more impulses, which will then propagate along the axon into the central nervous system. So the end goal, like I said, is the same. An action potential or several action potentials propagating into the central nervous system. It's just with receptor potentials in these separate cells, there's an extra couple steps involved. Does that make sense? So that's the way we classify receptors based on their appearance under a microscope. The other way, or another way that we classify them is based on what stimulus they detect. Most stimuli are in the form of mechanical energy, like sound waves or pressure changes, or electromagnetic energy, like light or heat, or a chemical stimulus, like a molecule of glucose, which we taste. Taste is an example of a chemical sensation. So looking at these different types, mechanoreceptors. Remember we talked about mechanically gated ion channels? They're going to have a lot of these. So these are sensitive to mechanical stimuli, like deformation. Deformation means deformed, like the structure is being deformed. Stretching or bending of cells. Examples of mechanoreceptors are the sensations of touch, pressure, vibration, proprioception, and our joints and muscles. And your hearing and equilibrium are actually examples of using mechanoreceptors. We have in our blood vessels and internal organs mechanoreceptors that detect stretch. Like when your stomach starts to fill, it stretches, we activate mechanoreceptors. Chemoreceptors detect chemicals. Chemicals in our internal external environment. We find them in our mouth, our nose, and body fluids. And they detect things like taste, blood carbon dioxide, pH of our blood, blood sugar, blood glucose, and smell. Thermoreceptor, activated by changes in our temperature. So there are the two warm and cold receptors, which we find on our skin. Nociceptors, remember I said no for pain. That's what these guys do. They respond to painful stimuli caused by physical or chemical damage to the tissue. And the sensation is pain. And then photoreceptors are found in our eye. They respond to light stimuli striking the retina, which is where you find those rods and cones, which are the photoreceptors. And then osmoreceptors are sensitive to water change, or osmotic pressure, basically your fluid balance. So if you can't remember osmotic pressure, what that is, it's just your water levels, basically. The third way to classify receptors is based on the location and origin of the stimuli activating them. We have what we call exteroceptors. They're also known as cutaneous receptors. You guys know what the word cutaneous refers to? Take anatomy, or have taken anatomy, you should know this. It's your skin. Okay? Which the skin is your exterior structure of your body, right? That's why they call them exteroceptors. because they're on the exterior of our body. So what do you think we call, or why do you think we call the next ones interoceptors? They're located interior, right? And our internal organs are also known as viscera, which is why we will also call these visceroceptors. Proprioceptors are the ones that we find in our muscles, tendons, joints, and even our inner ear. Exteroceptors, since they're located near the external surface of the body, are sensitive to stimuli originating outside the body, providing us information about the external environments, like pressure on the skin, touch on your skin or hair, pain, or just temperature around us. Intero or visceroceptors, located internally in blood vessels, visceral organs, muscles, and the nervous system, provide us with information about the internal environment. Things like pressure, which could be within like our blood vessels, what we call blood pressure, stretch of the stomach or blood vessels or intestines, many other places as well. Chemical changes, we detect a lot of chemical changes in our blood, like pH and O2 and CO2, and then hunger and thirst, which refer to blood sugar levels and water levels. Proprioceptors, as I said, those are in our muscles, tendons, and joints. Provide us information about body position, length of muscle, tension on muscle, position and movement of our joints. There are two types of these, tonic and phasic, providing us with positional information on the body or body parts while at rest or during movement. Table 16.1 here is the table that you'll find in chapter 16 of your book. Just gives you those various three ways to classify receptors, and then talks a little bit about them in greater detail, or maybe not even as much detail as what we talked about. So just kind of one-stop location if you want to use it to study. Might not be a bad way to do it. I'm not talking about pulling anything directly from, because we just talked about all those things. All right, I think we're going to call it here, and we'll start talking about this next, actually it'll be Friday next week, since we don't have class on Wednesday, and your test is on Monday. So we'll pick up talking about encoding simulation.